Hi, I'm Peter Cook. Welcome to the pan-genomic model, part 4. In part 2, I described a classical mechanism that drives DNA looping and clustering of the transcription machinery. And these clusters emerge spontaneously. In part 3, I showed that red and green clusters also emerged. So then, evolution has a choice. It can either spend energy to stop this clustering, or it can use these gifts, and I suggest it goes with the flow. In this section, I'm going to describe some properties of these clusters, which from now on I'm going to call factories and represent them as spheres. This is the dictionary definition. We originally defined a factory as a site containing at least 2 RNA polymerases active on at least 2 different templates. And this was to contrast it with this situation. A factory is equivalent to fashionable structures called condensates and hubs. I also believe they're equivalent to many other structures discussed earlier. And our kinds of factories are being subdivided into poised, initiating, and so on. So nomenclature is confusing. I'll make the pedantic point that our kinds of factories were initially defined by transcriptional activity in contrast to many of these other structures. Why would evolution want to have factories? Now, the concentration of the polymerase in the soluble pool is very low compared to the much higher local concentration in the factory. And this is going to act through the law of mass action to drive production of essentially all transcripts into factories. Some properties. I'll begin with a very early experiment where we integrated this construct into a mammalian cell that had a temperature-sensitive mutation. As the cell grew, this meant that the GFP-tagged polymerase was working normally. In the confocal, we got an image like this, where individual foci were not well resolved. This microscope has very poor Z-axis resolution, which can be improved by flash freezing without chemical fixation, and then cutting a thin cryosection. And then you begin to see hundreds of foci that you can count. And we also counted the number of active polymerases in the cell biochemically, and by simple division, we could work out the numbers per factory. In the electron microscope, we measured their diameters. Biochemically, we measured loops and found that they had a range of different sizes. We partially purified them and found that their proteins and nascent RNAs were the expected ones. So contrast this detail with what is currently still not known about condensates and hubs. For example, do they house a tiny fraction or essentially all of transcription, which we showed that factories did. We also demonstrated the factories specialize in much the same way that a Toyota factory doesn't make Teslas. An estrogen response alpha factory makes transcripts regulated by the steroid receptor, and NF-kappa beta factories make inflammatory response transcripts. And there are many other examples of specialized factories, and here are some early ones. Now, a central feature of our model is that the polymerase is transiently immobilized whilst it is active. And it is this feature that has prevented acceptance of our model, because we were all taught, and therefore know to be true, that a polymerase binds to a promoter and then tracks along the template like a locomotive. I've been going to international conferences for decades and asking this question, and I only get one kind of answer: Miller spreads. And here's a cartoon of a simplified version. But I want you to note that the transcript extends away from the template, and as you'll see in a moment, it is not entangled about the template. But this is what everybody recalls when they think of a Miller spread, the famous Christmas tree, where the branches extend away from the trunk and are not entangled about it. And it's easy to imagine from our textbooks how Christmas trees might be made. The polymerase binds, it initiates, a second one does, more and more accumulate, and we get the Christmas tree. And there are analogous images of lampbrush loops that you can visualize in the light microscope as opposed to the electron microscope that Miller et al. Used. But the arguments that I'll present apply to both. And there's going to be a very big but. So I'm now going to compare what happens when my polymerase transcribes this template. Whether or not the polymerase is fixed, a topological problem arises. It involves getting rid of the supercoils that inevitably arise on each side of a polymerase. It's solved by having a topoisomerase acting on each side. That's a problem that has a solution. But there's a distinct problem that arises if a polymerase tracks. Note that I've clamped the DNA and the polymerase is free to move. And then I'm going to show you a second topological problem arises that I will call the disentangling problem. And I think you will see that it must be solved. So watch. You can see that my polymerase is tracking along the helical template. And as it does so, the transcript is being entwined. So it has to be untwined. And you have to do it exactly the right number of times. Because if you do it once too many, you still end up with an entangled transcript. But there's no known mechanism to do this. But if the polymerase is held in a clamp, I'm now rotating the bolt so it's just screwing through the nut. The transcript isn't entwined. I've solved the problem, sidestepped it, and I'm getting an image like we see in the iconic spread. So now I'm going to reprise this movie that I've just shown you. We'll begin with a tracking polymerase, where the active site of the polymerase is going to follow a helical movement as it goes along the template. And when it's gone 10 base pairs, we've got one entanglement. And as it progresses, we get more and more until when we've gone 1,000 nucleotides, we've got 100 entanglements. Now that entwined transcript cannot escape unless you invent some mechanism to achieve it. And with many problems, you have a real problem. But if the polymerase is clamped and the DNA makes the helical movement, you just can screw the bolt through the fixed nut and the transcript doesn't become entangled, and you end up with unentwined transcripts, which is what is seen in Miller spreads. I'm going to reprise again and show you how the iconic image was obtained. Usually it involves one special kind of gene being transcribed by a special kind of polymerase in a special factory, the nucleolus. And so we imagine the promoter approaches a factory, it initiates in a polymerase. The fixed polymerase is going to reel in the template from the right as it pushes out the promoter to the left and extrudes the nascent RNA, all using the energy in NTPs. That now the promoter is still near another polymerase on the surface, and it might initiate. And again, and again. These 4 polymerases that I've shown are necessarily immobilized whilst they're active. And this is exactly what super resolution microscopy shows. So in the case of a human ribosomal DNA gene, the promoter is going to snake over the surface until about 125 polymerases have been loaded onto the template. On preparing the Miller spread, it's stripped from the surface and picked up on an electron microscope grid. Now recall that I'm discussing the only kind of evidence that is being offered in favor of a tracking polymerase, and I've said, no, it doesn't support it. Instead, it's consistent with the polymerase being fixed. Now, there's also a lot of other evidence for polymerases being fixed that I will not discuss here, but you can see the facts for that evidence. Finally, on this theme, I'd like to appeal to your common sense. We now know that the polymerase II complex is incredibly large. The enzyme is associated with RNA, RNPs, splicing factors, polyadenylation factors, nonsense-mediated decay machinery, and so on. We also know the nucleoplasm is very viscous. So which do you think is the likeliest? That enormous structure tracks through the glue, or that enormous structure is stationary as it reels in the template, so that the template, which has a much smaller cross-section, can snake through the glue, which we suggest it does. So to summarize, I'm calling the clusters of active polymerases that I've described earlier factories. I've argued that active polymerases are transiently fixed, and if you still refuse to accept this, you must find some evidence how one can solve the disentangling problem. And I've also said that factories specialize in transcribing different groups of genes. In the next section, I will discuss how regulatory motifs work. For much more evidence for immobile polymerases, see section 8. Thank you for your interest.